Thank you everyone for being here today. I know summer has started and it's so nice to see so many student faces in here today. And welcome to the second seminar of the Food Safety Consortium seminar series. And it is my pleasure to introduce today's seminar speaker, Dr. Jerry Lopez. He is an assistant professor and extension specialist with expertise in 4-H STEM at the School of Animal and Comparative Biomedical Sciences. Dr. Lopez got his PhD from the Department of Soil, Water, and Environmental Sciences under Dr. Chuck Gerber in, what was the year, Jerry?
Gosh, well, when I walked or when I actually graduated? 13, January of 2013.
Oh, 2013. And he joined ACBS in 2015, and he has a lot of experience dealing with school students and so on. He has 12 years of teaching experience in Tucson Middle as well as high school. And his research focuses on the transmission of human pathogens in indoor as well as outdoor environments, the risk of cross-contamination of food pathogens to humans, and more recently determining the occurrence of Cyclospora threatenensis in irrigation as well as wastewater in the fresh produce growing regions. And in addition to his research and extension, specifically in food safety, he has a very well established 4-H STEM extension program where he and his team have been working with Arizona 4-H staff to encourage the next generation of STEM innovators through culturally relevant STEM activities. And it is so nice to see so many students attend your talk. Speaking of that, today Dr. Lopez is going to talk to us about some of the work he's been doing with Cyclospora threatenensis and also the importance of working with stakeholders. I mean, produce industry is really important to Arizona agriculture. And he has been working with the stakeholders, and he's also going to educate us on that also. So,
okay.
I leave it to you, Jerry.
Thank you very much, Sarna, for the opportunity. Thank you all for making it today. Some of you guys drove across town to be here. So hopefully you'll be able to gain some insight, particularly some of my collaborators out there, and some of the results of the study that we just completed. It's an updated version from the last one that I sent you guys. So Cyclospora outbreaks and importance of working with stakeholders. The plan is to go ahead and weave through the discussion, the connections of how it's related to working with partners in industry, and the lens that I think it's important to gain or to have, even as researchers, as faculty, as we're working with industry and the community. And not just necessarily from a research perspective always. So let's see. All right, so Cyclospora parasite outbreak. What you must know. Do any of you guys remember any time recently or within the last year or so? What was going on? What was happening? Did anybody hear anything about Cyclospora, or was it only some other famous pathogens like Salmonella and E. coli? Right? Did anyone here? Can anyone sort of have an idea? Yeah, what did you hear about?
The E. coli outbreak at Chipotle.
Okay, yeah, definitely E. coli got a lot of attention, but Cyclospora is trying to make it up there. It's auditioning for some really good spots. So did anyone, what was that? Did someone say something?
I was saying Cyclospora is competing with E. coli.
Yep, it is definitely, and Salmonella. But so did anyone hear any cases or some news on? I know some of you are nodding your heads. All right, so I know we all like to eat out, right? I think most of us at least eat out once a week, if not more. And we try to maintain our healthy eating habits with some lettuce. But okay, let's go ahead and take a look at this. So before we move on to these, this is just an outline of what the topics are going to be. I want to give you some background on what Cyclospora is. right? Some classification, symptoms and treatments, the distribution, and then a little bit about the life cycle. And then I want to move into the C. cayetanensis, the outbreaks in the US. And then finally, I want to wrap up with how it relates then to a project that we just finished being funded from CPS after 2 years. And through that, I want to go ahead and incorporate then, like I mentioned, the importance of working with stakeholders. So what is Cyclospora cayetanensis? So it's a human protozoan intestinal parasite, right? If you notice here, which one does the red one point?
I'm not sure.
Yeah, I'm not sure. Okay. So you can see we have Giardia, Cyclospora, Cryptosporidium, crypto being the smallest. Cyclo then is about 8 to 10 micrometers in diameter. Causes an infection called cyclosporiasis. Really, it comes down to, if it's an intestinal infection, what do you think we get? Diarrhea, right? Definitely we're going to end up getting some diarrhea. So people become infected after what? What ends up happening? If it's an intestinal thing, what do we need to ingest? Food or water, right? And if it's an intestinal pathogen, what needs to, that food or water needs to be then what? Contaminated with feces, right? Exactly, needs to have feces in it. So that's the connection that sometimes Cyclospora, a lot of people think it behaves or acts, has the life cycle of a bacteria or a virus, but it's completely different. And it really surprised me when we were in Mexico giving a presentation on the topic, and a lot of the industry workers, again, it's just that understanding. We sort of are focused on bacterial pathogens, not understanding exactly where Cyclospora comes from. And if it's an intestinal type of infection, then there's definitely a connection there of where it's coming from. And being able to educate then our partners from that perspective, I think can really help us in this next season that we're already in, practically. So one thing to keep in mind is that Cyclospora is not spread directly from person to person, okay? Unlike these other pathogens. So some classification, it's definitely coccidian. It's related to these other types of parasites, Toxoplasma, Isospora, Cryptosporidium. An interesting fact that I think it was really important to indicate here, Dr. Inés Ortega, and we're all familiar with Dr. Sterling, right? Does anyone know the story behind this? Would someone like to share? Kelly, do you remember, or do you sort of? Yeah, well, you sort of saying Pat.
Dr. Ortega was in her, she got a degree at the University of Arizona as well.
Yep, exactly. So that was really interesting. So I'm in a meeting. I hadn't really, I've read up on it and I had some, the background information on it, but it was really interesting to be in a meeting, finally meeting Dr. Inés Ortega. She's now at the University of Georgia. And it turns out that she was Dr. Sterling's student. And then at the time they were working on a project, they ended up naming Cyclospora cayetanensis after the Peruvian university, Universidad Peruana Castellano Heredia. So it was really neat, and she actually attended school here, so it was interesting to see that. One of the things that I think we might end up having a discussion later, once we start looking at these other similar organisms, because they're very similar to Cyclospora cayetanensis, that presents some challenges when we're actually doing some molecular work. And that's where as we're working through identifying Cyclospora as a true positive, this is where we're faced with those challenges. Sometimes unlike what happens with other organisms. Okay, so some symptoms and treatment. So once you're infected, and how are you infected again? How does that happen? Ingesting what? Food or water, right? Okay, or whatever that food might be. And many times if it's ready-to-eat meals, such as lettuce or cold cuts, those type of things, Thank goodness there hasn't been anything associated with a cold glass of beer, though, right? So at least that we know. So it's usually occurs about 1 week after exposure. If not treated, symptoms can definitely last several weeks, up to a month, okay, or even longer. There's even been cases of relapse, they've noticed, okay, where someone will end up getting actually sick, they start getting better, and then they fall back into that cycle again. Some people infected are asymptomatic. So what does that mean for those of you undergrad, graduate students? Okay, yep, exactly. They don't show the symptoms at all. Now, that becomes a problem or a big issue when? When do you think that really becomes a problem? Is that a problem for us here? Workers, exactly. And particularly workers that are in close proximity to fresh produce, that's where it really becomes a concern. Symptoms may include the following from watery diarrhea, loss of appetite, the cramping, fatigue, low-grade fever. Nausea is less common. Diarrhea is the most common symptom. And as far as treatment, you can treat it by this combination of antibiotics, which is the common names are Bactrim, Cetera, and Cotrim. Thank goodness people don't die from this as long as you stay hydrated, at least in developed countries. So again, it's recommended, just like every other illness, plenty of rest and staying hydrated. And this gives you a little idea of what the sporozoites and the actual oocysts look in the different stages of this organism. So I think if there's someone in this room that's probably most interested in this geographical, how many of you guys travel so much? So for those of you that travel quite a bit, I'm sure this is one of the things that you're definitely aware of and keep at the forefront of your thoughts when you look at it. It's definitely most common under tropical, subtropical regions, anywhere from Brazil, Chile, Guatemala, Mexico, and so forth. You see the actual distribution of them. And some of the produce that's been associated with that in previous years with outbreaks has been raspberries, basil, snow peas, and then the mesclun lettuce. And most recently, there's been some association with romaine lettuce, but again, it hasn't been identified that was the cause of the outbreak. There were samples that were collected positive, but yet they weren't connected to the actual produce. Now, why is that important? Why is it important to get that straight? Again, talking about the importance of working with stakeholders. Exactly. Those growers, that entire industry doesn't want that, but yet, what's the actual connection there? Why should us as researchers or young scientists, why should we be concerned with what's going on? Don't we just want to go ahead and find who the culprit is or what caused it or where it started?
Prevent it.
What do you mean by that?
Well, if you know it's there, it's a potential for a disaster to happen, and now they can take action before that.
Exactly. And I think it's real clear, though, in terms of as we're articulating, the difference here is to make sure that we identify, okay, yes, it was identified in existing produce, but that's completely different when you actually have a product that's on the shelf that's been, the traceback study went back that this individual was sick from that package, and that is associated back to where it was. And so I think it's important for us to make sure that we keep that in mind. And then as young scientists, I think it's critical as you identify the research that you're doing and who we're working for. It's almost like two parts. We're in the middle. We're the bridge between the industry and the partnerships and also the science that's out there. But we have to be sensitive to that fact and recognize the importance of building that partnership. Okay, so the life cycle. In a nutshell, just as you pretty much can describe, you go ahead and you need to ingest then oocysts that are sporulated. So if they're sporulated, that means then what? Oocysts can occur in what form then? You have sporulated oocysts and then unsporulated, right? So what do you think is the difference then already? Again, thinking back to this organism and the difference that it is between others. Think back to a bacteria. Do you have an infective stage of bacteria? Does it become infective at a certain point? A pathogenic bacteria? No, right? So here, this parasite is a little bit different from that sense. So when it's shed out of the individual, right, the person that's infected, it actually is unsporulated, meaning that it cannot infect. It's not infective at that point. Okay, it needs to be in the environment, and this is where there's some short studies that have been done to identify that it takes 7 days, 14 days, the temperature, there's an adjustment there. So the actual organism is able to go ahead and meet a mature sporulated form, and then it becomes infected. Since we're on that topic, again, knowing what you know about it now, yes?
Is that considered the sporulation phase an asexual part of the life cycle?
Not at that point. Not at that point. At that point, it's actually they're not mature sporozoites. There is a sexual and asexual in the actual epithelium and the intestinal infection. Yep, there is.
I'm just curious, are there anything specific requirements of these spores to germinate? Like if they fall on meat, they may not pernicious substrate, but carbohydrate-based substrate might be more conducive for germination.
That's a good question. I don't know if someone out in the audience might know something more specific about that. If it lands on certain types of substrates, is it more conducive? I know they're very sensitive to desiccation. That indeed it is, but in terms of...
You said they're sensitive to desiccation?
Yes.
But the spores should be more resistant, right? Don't they share?
Yes, but and good point. It's all relative. It's relative. In terms of heat, in the field or in an environment where it's direct sunlight, the studies have shown that it's actually more sensitive. But in terms of other types of treatments, such as chlorine, they're very resistant to that. But again, going back to this, then, if you start understanding this organism, then, and as if right now we're going to take a look at some outbreaks to try to understand how this outbreak occurred and where was the point of cross-contamination is really important, because if it's not person to person, a person can be asymptomatic and shedding, but yet it requires some environmental change and it requires a duration of it to be in the environment before it becomes infected. And then we can go ahead. So with that in mind, I want to pose this question. How do we determine its infectivity? Do we just go ahead, Monique?
Human volunteers.
Human volunteers, huh? Can we sign you up?
You can look into, you can grow artificially and just look at whether they're multiplying or not. Even by simple PCR-based technique, looking at the copy numbers and so forth, that if they are under certain conditions, if they're multiplying or not.
Yeah, that's a good point. I mean, you raised some really good key points there. Are they multiplying? So can they multiply in the environment is one.
Are there virulence factors?
Okay, like such as?
Is there a particular component that if present, a toxin would be there? I mean, is it related to that, or is it just an infectious dose? There has to be a mass.
Yeah, so my understanding is it's an infective dose. There aren't, at least based on the science that's out there right now, there isn't a toxin that's associated with that. It's the actual, the immune system responding to this infection of these worm-like sporozoites that have been infected into our epithelial system. So, but that's a really good question. So that's something that hopefully, I mean, keep this in mind as our discussion continues. can they multiply? But going back to the original question, how can we determine if it's infected?
And humans are the only hosts, right?
For this particular Cyclospora cayennensis, it's only humans. There are some similar organisms, some other species, and their family with the Eimeria family of similar organisms. There's some other strains of that or other species, but not, this one is only affected to humans. Yes. Okay, so how would you?
Grow epithelial cells in liquid medium and add different concentrations, I guess, of the sporozoites and see if they're causing the destruction of the membrane.
Okay, I mean, that's a logical, good explanation, or at least a good study to see if that's potential in terms of us replicating the environment in the intestinal tract, right? And to see if that's possible.
I was just thinking, like, if you have one, and I'm thinking about the farm level, you have one worker who's symptomatic but shedding, and if you see some additional people getting infected among the farm workers, before it reaches, it takes, I don't know, 60 to 90 days probably for the crop, most of the crops to harvest, and then another few weeks before it gets into the consumer. And so if you see some additional workers getting infected, so basically monitoring the health of the people working in the farm would be.
That's a really good point. So if there's testing going on, surveillance going on of either the hygiene stations, swabbing in the area of different points of contact, probably hand transmission or other areas around the porta potty. I mean, that could, and if it's identified there, then being able to keep a surveillance of them, then that's a really good point. I know there's some industry partners, and I'm not sure if you'd like to share a little bit, Robert, or that perspective of it. I mean, I know there's some industry partners that do some surveillance.
Yeah, we do, but it's mostly for enteric pathogens because those are the ones that are most likely when we do risk assessments. We're looking more for Salmonella, E. coli, the usual suspects. But the big deal is that there aren't readily available methods to quickly identify, in this case, protozoan parasites, not just Cyclospora.
Yeah.
More likely than hand transmission or an individual worker getting sick is either endemic in the environment or especially in the water. In 1995, the raspberry example that Dr. Lopez had up there, a farm used canal water for their spray dilutions, and it was loaded with Cyclospora. So they basically inoculated every berry in that crop with their spray applications.
Interesting. So yeah, so going back then, I mean, think about it a little bit, again, in terms of what's actually, what can be used to determine the infectivity. Definitely that's one. So once it becomes, through that process, as it's becoming sporulated in the environment, there's cross-contamination, right, with water and fresh produce, whatever it might be. It's ingested, and that's when the cycle continues. There is an asexual and sexual merozoites and different stages of the merons occurring and in the zygote of a fertilized form, so there's some diversity there. So here raises another question. Because there's some diversity in terms of zygotes, what should come to your mind then with that? So we're talking about Cyclospora cayennensis.
Other species there, and there's a
or serotypes, right? Different serotypes from different regions. Again, those are some challenges that this organism has. So researchers are really challenged with this in terms of being able to identify if there's genotypes. And there is some work currently being done. There was some work that was published from a group in China that's looking at mitochondrial DNA as opposed to the 18S for serotyping for sequencing. And I know the CDC is out to publish some work also with the mitochondrial DNA. What we've been doing is, again, we've used a target ITS2, the interspacer transcriber 2, that's being used. And we developed a TACMAN probe with our collaborators from Japan, along with our group here at the West Center with Callie and Walter and Chuck Gerba and the rest of the group. But again, and so far, I'll share a little bit how it's been successful to a point where we're at in our stages, but it's definitely different in terms of what's going on with now them noticing that mitochondrial DNA might be a better target because being able to differentiate between genotypes.
So before you move on, I was just wondering, is there any in vitro culture system for this or no?
You know, unfortunately, there isn't, because humans are the only ones, unless someone else might know of something that's currently, I mean, what you mentioned in terms of epithelial cells, epithelial cells can be grown on petri dishes, and that's mimicking the gastrointestinal environment. I mean, there is a potential because we have oocysts. Oocysts can be isolated. Did you have a comment, or?
That actually brings me to another question. You said Cyclospora belongs to AB complexa, which the malarial parasite also belongs to. So, is there any kind of similarity? Would you see Cyclospora in the blood?
Not that I'm aware of, no.
Just in
Yeah, it'd be an intestinal epithelial type of infection. Yep. Yeah, it's definitely, and it's probably in a different family from the other organism. Okay. All right, so now moving into some outbreaks. So these are the outbreaks that we witnessed in 2018. So one of them was the Del Monte. I don't know if you remember the actual fruit trays, excuse me, not fruit trays, the veggie trays that were up in the Midwest area. We had, as of September 2018, it's pretty much what it was, 250 cases. There was 4 states. It was somewhat small, but yet it was definitely beginning to come up and surface. There was a recall on some other items that also had that really impacted, you know, lettuce wraps and a lot of different types of wraps and salads that contain different types of, you know, meat in it. So this was one of the outbreaks that occurred. Now this one here is definitely a much larger one, right? When you really think about it, we had 16 states, 511 cases, quite a bit, a whole lot more. So when you start looking at these type of outbreaks, who are the players? Who are the stakeholders in this? I mean, we're definitely part of it as researchers, academia, but who would be the other ones? Absolutely, the producers to start with this is the foundation of this aspect, right? Yes, Dan.
So there's these outbreaks were a week apart. So were they the same or the same organism?
Oh no, so they were actually occurring for a while. I updated. This was the most the last updates that they had at the time when I prepared this.
Okay, so they're not. It's not the same organism affecting different products.
So yes, same organism. Cyclospora cayetanensis was found in 2 different areas. One was these veggie trays, okay, and it actually the salads. And if we notice some of the states, let's see some of the states that are there. Yep. So there was a total, if you look at these, 250 plus the 511, about 750, 761 total cases, right? So who are going back? So academia is definitely part of this, but in many ways, we don't become a part of it until when? Pardon me? Consumers, or how do we, I mean, we're definitely dependent on funding. It's not like we're gonna go out there and go to McDonald's. Can I go ahead and sample? You know, can you start sending me samples every day for a month? No, right? So who else then? Who's the actual processing plants? They are, but then let's clarify that a little bit. So the processors are, but who's doing their testing?
Third party.
Third party, right? Third party. Some of us might be fortunate enough to have, a testing portion that we could go ahead and start doing some sampling from that aspect of it. Who else then?
Regulatory agencies, because they have to investigate.
Absolutely. So which ones are those? CDC,
FDA.
FDA, they're definitely then becoming involved, right? Just like we saw it with E. coli and Salmonella and these other outbreaks. So we start getting different people involved with this. Now, should we all have the same agenda? We should ideally have the same agenda, right? And I think we all have a common goal. Do we approach it differently though? Are there external pressures on the decision makers? Absolutely. And I think it's that aspect of it though, maintaining a certain lens and focus of where we're in this together. If we go ahead and approach it from one angle because of the pressures that I have, but that's really going to impact another industry. I mean, we need to maintain what? What do we need to maintain between all these stakeholders? Communication, absolutely. But with that communication, you need to build trust. Consumer confidence, trust. That consumer confidence is dependent then on what's being sent out there, what's being publicized, right? And who's coming up with those ideas? Well, it's the regulatory agencies, it's interviews with faculty, with growers, producers, the industry. So they all really make a major impact on this. And so this is where I think it's critical for us as we're approaching these type of challenges that we keep in mind, definitely communication, but we have to maintain that trust. Each other's bottom line interest needs to be everybody's interest to a degree. And then there's a debate, there's a dialogue that occurs. Well, wait a second, at what point do we need to then assure this in order to move on? But I think if there's a good consensus between the entire team, those type of decisions can be approached in a very positive way, in a fruitful manner, that everybody benefits from it. So now, what do you guys think about this? During that same period, though, there was 2,299 cases. How many did we say that those two outbreaks had? Yeah, 761. So what's going on? Hospital, yeah, these were all laboratory confirmed. 33 states. The other one had what, 4 and 16, and the 16 probably had the 4. So this is almost double. So what can you actually get from this then?
This case number is looking like this bimetal car. And how do you know, I might be in another part of the world and I got it and come here and being sick. Is that number included there?
Good point. No. So these actually, an excellent observation, Arun. These right here are actually non-international travel. So the CDC does a great job in differentiating international travel cases to domestic, and so those are strictly non-international travel, all domestic, and these were also the same thing. Same period. So not even what, almost maybe a third, a little bit less than a third of all the outbreak cases were associated with 2 distinct outbreaks. We had almost 1,800, or somewhere around 2/3 of that number then were associated with non-outbreak. So what could be happening there? Okay, potentially, definitely other foods, and I think cilantro and basil were strongly associated with these other ones. But what becomes so difficult when you're doing a traceback study? Someone ends up getting, do you guys remember what you ate a week ago? I mean, it becomes really difficult to trace back and see where that center case was, and identifying where those cases are actually coming from. And then cross-contamination, if depending on their hygiene You know, I think all the possible avenues of a cross-contamination are really broad. We sometimes right away want to focus that it's just a product. But remember, if someone becomes ill and they're in that environment, if they're working in a restaurant, I mean, based on their hygiene and based on, you know, the regulatory aspects of what they're doing there, can there be a cross-contamination? Absolutely. And that could end up sprouting another event. But yet everybody's focus is over here on this other scenario.
But looking at the case, the bimodal distribution and the dates below, which I can't read accurately, but March and July something.
Mm-hmm.
So is that coincides with the harvesting period of certain crops that is there data like that? You know?
Oh, that's a good question. I'm not
Trying
sure.
to see without having all the information, trying to make some national
Mm-hmm.
argument that where it might come from, and then perhaps it's coming from certain crops that are harvested on the time
Mm-hmm.
in
Well,
certain region. Of
our study that we just completed did find what we learned from the results was that definitely there wasn't a seasonality associated with Cyclospora in terms of its occurrence.
that was not.
There was not. We do see a seasonality of the season throughout the year that infections begin to occur, right? We definitely end up seeing that, and there seems to be a relationship with fresh produce in some fashion, but we still don't know exactly how that's happening. You know, because over a 2-year study, 2 different regions, we didn't see that there was a seasonality of these infections.
You know what? I would speculate that today, because you know, I can grow cilantro today, a lettuce, and I can store it, and then I'll eat it after a few months. So it becomes almost impossible. Had there been no storage event, if you eliminate that
Mm-hmm.
food chain, then cilantro is available primarily during the summer months and the winter months. You know what I mean? Like
Mm-hmm.
you, and that is being broken because of
Mm-hmm.
this practice. Basically.
Yeah.
I know you said it only affects insects, but can wildlife carry this and spread it? I mean, they're asymptomatic and it obviously doesn't affect them, but can they carry it and spread it?
You know, I know we have a couple of experts here on this, and I know there's been some what we definitely know is that there isn't the same Cyclospora cayetanensis will not infect, you know, dogs or deer or mice. So it's not going to do that. Carriers, though, that's a different question.
I mean, the feral swine carry the pathogenic organisms into some of these produce fields, or the deer did, and so I guess that would be a question like that.
Now,
Even though they're not infecting them, they very well could. I'm curious as to whether or not they could.
do any of you guys?
I don't think it's really been like that. We wanted to do that.
Mhm.
We haven't had it on before now. Because we were thinking the same thing. Maybe it might be present in animals.
As a reservoir.
As a reservoir, yes. And but we haven't had the opportunity to look into that.
Yeah.
That'd be a good thing.
Did you want to add anything?
No, I was at a meeting at FDA about a year ago, and that was identified as a knowledge gap.
I wrote a paper recently, and they looked at the specific type of like mussel shellfish, and they looked for Cyclospora in the shellfish because they filter out like so many gallons of water for like over a period of time, and they found in the gills that there were like Cyclospora like DNA, so like they're able to filter it, and it doesn't necessarily affect them, but it just like accumulates maybe.
Mm-hmm. That's a really good point. So there's,
So I have a question.
Sure.
I know Dr. Dar mentioned that, you know, we are storing these leafy greens over a period of time. Has anyone looked at how long do they survive? You know, let's say there's storage under refrigeration conditions. Does the organism survive for that long, or how long can it survive?
yeah, exactly. I'm pretty sure the literature has shown that it definitely can survive in a good environment where there's plenty of moisture and you know, it's conducive to that environment. It could definitely last for a month. The literature has indicated, Kel?
Have they done like DNA assays to determine viability, or how are they determining viability?
That's the thing. They only goes back then to this. what do you guys think would be the most, or not that it's easy, but what tool do we have to be able to determine viability? Microscopy. That would be, but can you imagine the stages of an unsporulated oocyst, the days 7 to 10, 12 days, depending where that range would be for it to become? This is why it's been real challenging to do, disinfection, what type of antimicrobials are sensitive or effective against them.
And has anyone looked at natural type of antimicrobials?
I haven't seen
I know
anything.
chlorine is pretty resistant, but
Yeah, I haven't seen. But then the challenge though is, I've been approached, to do these type of studies, but how do you go ahead and determine the viability if you do the dosage? My first instinct was, sure, let's do it. But then as we start analyzing, oh, wait a second, how are we going to actually determine the viability of this?
So I have a question. It kind of connects with the discussion, but I'm thinking of previous slides. You said that they treat this disease with antibiotics, right? Which
A
is
combination.
Kind of, okay, combination. Has anyone looked and seeing that this parasite has bacterial symbionts that perhaps are the ones that confer the infectivity or virulence of the protests?
That's a really good point.
I'm thinking with my mind and the
Yeah.
system that I study. And
That's a really good point, because why would they then be sensitive to that?
yeah.
Yep.
Just do you have some of the spore material, the Cyclospora, it is spore with you here in the video?
Yep.
Okay, give it to us. We'll run in 16 minutes and just answer the question tomorrow.
Huh. That's a good point. That's a good point.
We have it. We have the universal one. Just run.
Ah,
And
interesting.
This will be. Yeah, I have a way to isolate this kind of DNA. You can give it a try.
So that's a definitely discussion. Let's have that discussion. Yeah, we ended up part of the things for.
No, I'll say something. Because we work with another parasite, which has some at least morphological similarity, and we try to see whether they're germinating or not. But I was thinking, if you have spores, perhaps you can put them into different substrate, raspberry, candidate substrate, and keep under different in the incubator, different conditions, and just look under the microscope to see if they're germinating or not. That could be very
moisture.
Yeah, different parameters. That could be, and that has ramification with respect to managing them in the farm.
I know Dr. Ortega, and there's some other groups that have done some work on the sporulation process of it, and but again, it's the antimicrobial, not having a human model or not having a model that we can go ahead and animal model has been the challenges for this. But
there's no other mammal, or there is no.
Nope, there isn't, at least up to this point.
Oh,
Monique's pretty interested though in developing one.
Yes, I know.
She almost showed up with a little chicken the other day. So again, I think I hope I emphasize the importance, or at least giving you a glimpse of how the different stakeholders, it's important to be able to work with them. And even though, again, we might have different emphasis at the time, we need to definitely compromise to a point and not jump the gun, to go ahead and, despite of what pressures there might be. Because I think it's like we've all experienced this. You could do 100 things that are great, but it's that one thing that you do wrong is what's going to stand out.
So what are the best practices that you're going to tell industry to manage this risk, to reduce it?
It's basically the best practices right now, the hygiene best practices, good management practices. It's what's the same ones that are being utilized for other pathogens. At this point, it really is. If anything, though, what's unique about this is that it's definitely an intestinal one. So I think that transmission route needs to be looked at, I think, examined a little bit closer. Because when you really think about the volumes and volumes of water that's out there, that even though there's been cases that there has been, you're basically contaminating a field through, the overhead, watering, right? Irrigation like that. But I think in overall, when you really compare all those approaches and knowing that this is an infection that occurs with humans solely, I think that definitely needs to be looked at a little bit closer. So building trust, working together to solve the complex challenges. And again, we could definitely add to this, but I think, like Kelly mentioned it, having the communication from the very beginning, being transparent about this and understanding their lens. I need to share, we were working with some stakeholders in this growing region, and I definitely had my perspective and even the study design of the way we were going to approach this study. But after talking to them and hearing and sort of digesting their view, and as we were then preparing the report to submit, then it sort of came out of the surface from that. It became apparent that the way we were presenting the data did lean itself to a certain direction that wasn't beneficial to all the stakeholders. But it was very easily, you could go ahead and present the data in a different fashion, still identify its occurrence, and look at the way it's impacting, or the positives, negatives, and where it's found and those type of things, but yet in a different perspective. And I think working with our group, we were able to sort of see that, and it became really important. So, okay, so real quick, I know we're obviously there's an industry need because of the knowledge gaps. This is now we're transitioning into the actual project that we completed. This was back in, a couple of years ago when we submitted the project. We were looking at two different areas, irrigation waters and then also wastewater treatment plants. Now, something that we wanted to sort of identify if there was a correlation between Cyclospora and wastewater treatment plants to irrigation water. But again, going back to that genotyping, that doesn't exist yet. There's no way to really do that. And so there's definitely a gap there. Now, this other idea of how the canals or irrigation waters are becoming contaminated, again, that's a whole other specific problem. We weren't able to identify that. We were able to determine if it's present, but yet where it's coming from becomes a whole other challenge. And I think there again, working with our partners, that becomes more of a detailed type of discussion with our collaborator from that region. So that way to determine what exactly, where were the positives at, what's an association to that, and for there to be a closer examination at that level. So again, as I indicated, it was definitely part of the RFP. The objectives were determine the occurrence of Cyclospora cayetanensis in irrigation waters, as well as in wastewater treatment plants. There was 3 wastewater treatment plants in one region and then in the other, both Arizona and Texas. So here's the method. As I was putting this together, I think here's the other side of the lens though. I think our industry partners sometimes, and perhaps some of our funders, do not really understand the amount of work that it takes to go ahead and get this done. The manpower, not only the manpower, then the actual supplies that are involved to get this type of work done. I mean, when you're looking at from sampling, and then we need to we have that set budget. If there's a delay, if there's an adjustment, we're not going to be able to get on the phone and ask for, an additional amount. It's set. And so we need to be able to manage that. So I think the compromise and the understanding definitely, bridges across both sides. So we ended up collecting 100 liters, up to 100 liters of irrigation water. The first picture up there indicates that. And then we had 1 liter grab samples from raw sewage and then the treated effluent. From there was 2 different processes. for the elution and the concentration. The EnviroCheck filters were actually eluted and concentrated using that shaker machine. And then in terms of these 1 liter samples, an HA filtration process was used to capture the oocyst. And then from there was a concentration step. And then as you see in this picture right here, then those are centrifuged, the pellets are centrifuged, and then resuspended in an aliquot. And then we end up aliquoting that amount, depending if it's, anywhere from 5 mL to 15, depending on how big the pellet was. We'll go ahead and have some stocks that end up being stored in some other containers in the other tubes. Now, real quick, what challenge or problem do you think could occur by putting your 15 mil or 5 mil sample into aliquoting it? Contamination, okay, that's one, but more in the sense of dealing with your target, with your oocysts, right? Less number or nothing at all, right? We've tested some. They were positive. We run out of that one. We start on the other one, and the chances that aliquot, that 1 mil, is carrying that oocyst. So that also presented some challenges. But anyway, then we end up having the extraction, DNA extraction. Now this process here was not the FDA approved or recommended, suggested method that came out, a year ago. It was published literature, that was reliable, that was used by CDC and other labs for DNA extraction for Cyclospora. The EnviroCheck was definitely, that's a filter that's been used for Cryptosporidium and Giardia and it's an EPA approved method. So it was a combination of these because there wasn't an actual approved method or recommended method at the time. And it's the same thing went with the ITS2 that we opted for that model. Yes.
So Jerry, when you are taking the samples, you say that you take aliquots, of course, because you can sample everything, but is there any way that you can concentrate your sample?
Yes,
so you can't, the spores or.
That's a really good point.
Sometimes, for some parasites, you do a sugar flotation technique that brings the spores or the parasites to the surface and then you have some protection.
Yep. No, that was actually an area of funding that we submitted a proposal to CPS. Unfortunately, we weren't funded for it, but that was actually looking at that. The challenge of concentrating, because it's not like a stool sample. It's going to be there. Environmental samples, the concentrate's going to be. So we're looking at, different approaches to be able to do that.
I'm going back to the sewage. Is that human waste and livestock waste or just human?
That's a good question. It should be mainly human, but if there are canals, not the canals, but if the drainage areas in the town, the community, or actually has all rain runoff, that's a good point.
Jay, if I just a quick note, as you mentioned, last couple of days I was in Oregon State, from the biomed department, biomed science, and there's one gentleman who works on environmental DNA samples, eDNA. And what it does is basically same thing, concentrate the sample using nitrocellulose filter and whatever size, and generally and then one thing he did which turned out to be quite useful, how nitrocellulose is made, basically dissolve the entire thing in acetone. Instead of trying to isolate it, that dissolve the whole thing.
Is that right?
And get the DNA of the whole.
Wow.
So something.
That's interesting. Yeah, this one is a 1 micrometer pore size, so it's by size inclusion. If you remember, the size of Cyclospora was 8 to 10 somewhere. Definitely going to go ahead and capture that. And then at the end, we go ahead and we'll do the qPCR run, right? Moving on then. From there, what we ended up doing, once we had the presumptive positive at that point, we then went ahead and did the conventional PCR using the same ITS2 primer that we had. And we went ahead and ran a conventional and sent it off for sequencing. Now, we only did a subset of these, particularly when we first started and we were optimizing and ensuring that it was hitting our target area. And from there, again, the great work that Daniela and some of the undergraduates did, they would then do the NCBI blast, and we had some really good confirmation on solid confirmation of the ITS2. Now, some additional things that we went ahead and we did there with our collaborators from the FDA, the sensitivity assays, we went ahead and we looked at some oocysts that came, or DNA from the CBC, to make sure it's sensitive, so it is hitting it. The specificity assays, there was some work that was published, some work that Alex Da Silva and Helen Murphy from the FDA did, and they went ahead and hit a different, like 11 pathogens that are very similar. And so they sent us about, I want to say 6 of those. So we went ahead and we ran them against that, and we didn't have any hits, which were good. We're in the process of getting a BEI, a second or a third party, I guess, organization that allows you to get some DNA, certain pathogens from there. Or one of the 6, I want to say, was Imeria, which is then really similar, to the Cyclospora. So we're hoping that we're going to be able to get the remaining ones, or at least a few of those, from this other organization, to make sure the specificity. Yeah.
So since there's a limited detection there, is there also any quantitative methodology that can be done to find out?
Besides, with the exception of microscopy, of being able to, and again, that's like the gold standard right now, that's really preferred to go ahead and confirm it. I think that's why I'm hoping that the mitochondrial DNA genotyping might be perhaps become another. I'm not sure if you've heard anything different or. Yeah. So, but yeah, the microscopy is the only way. But it's an old art. And again, hopefully we'll be able to have some funding in the future to go ahead and have one of those microscopes that can actually, identify it. But so this is what our protocol was and the development of that assay. Hopefully we'll get it out here in the next a month or so. So our results overall, we also had E. coli and total coliform samples that were taken at the time. But I want to focus, particularly because of the sake of time, with the gene copies of Cyclospora. So if you see canals, the average gene copies per liter were about 29 gene copies. So in this particular region, in Arizona, there was 15 out of the 119 samples, so almost 13% were positive. I think at this point we're feeling comfortable that it was positive as opposed to presumptive. But even if presumptive, we have to sort of err in the caution side, that there's the potential there. But again, does this address viability? Does this address infectivity? It doesn't, but I think looking at it from the lens that it has the potential of doing that. Now, if you look at the effluent and the influent wastewater treatment samples, influent 55%, 46 out of 83. As opposed to the effluent, there is a reduction, 36% positive. Okay, that one was region 1. Region 2, again, 5%. We ended up taking a considerable less number of samples there as opposed to the other region. Our collaborator over there ended up moving. There wasn't a replacement there, so we had to go ahead and make up that difference in our other region. But again, very similar. We have a really high positive on the influent, indicating that it's definitely present in the community. The effluent, even though there's still a reduction, it's still present there. Now, is it getting into the watershed? Yes. Is it getting into the irrigation systems, that's where it's debatable. And with the terms of the genotyping and the lack, that data gap there, it's hard to determine if it's similar. So some key outcomes, again, Cyclospora was found definitely in irrigation waters, 5 to 13% range of the samples. Okay, it was definitely found in influent water, 46 to 55% between the two regions, and then the effluent, 26 to 37. So the key learnings, though, I think is what's really important to walk away from here. We definitely didn't see no seasonality pattern was shown for the occurrence of Cyclospora. Okay, when you look at the actual seasonality of, hitting it, there wasn't anything. No correlation between E. coli and coliform data for the occurrence of Cyclospora. Okay, that means that's why in our current study that we have, we're not monitoring for coliforms. These findings do not determine the viability and effectivity of the positive samples. So I just want to definitely point that out. These findings indicate that there is an incidence of C. continence infection. This is what's really interesting. I think this bullet here, that because we see it in the influent and the effluent, so we're definitely, it's in the community. The infection among both communities, it's present there in very similar proportions in terms of number size, the samples that were taken. So going on to the next bullet, no determination was made of potential source of contamination of Cyclospora cayetanensis in irrigation waters. So again, when you look at open waters and all the different points of, human contamination can occur, and so I think there's still a lot of data gaps that are in that area. And I think working together with industry, the stakeholders, can help improve trying to monitor where this is actually occurring. But I think at that level, it becomes something more at that community partnership, not necessarily, and maybe we can extrapolate from that in the bigger picture, but again, keeping in mind the importance of the interest of our collaborators in those communities. And then finally, these findings also suggest that persons involved in the production and harvesting of fresh produce and who live in these communities might pose a potential risk through some cross-contamination pathway. Yes, Cal.
So one thing you might point out, Jerry, that there's these canal samples were taken from areas near the wastewater treatment plants. If we didn't actually include canals from farther away. So that 5 to 13% might be in those areas, but maybe if you're looking at, for the downstream areas, those numbers would be reduced quite a bit. So I just don't want people to think that it's 5 to 13% in all areas now.
That's a really good point. Good point. Good point, because the sites were identified around the wastewater treatment plants and then irrigation canals. Even though there was no direct connection between some of the wastewater treatment plants were releasing it into the watershed, others was reclaimed water, others were being stored in actual like pumps and different for a reservoir type of. So, but yeah, good point. Okay. So what's next? I think based on our findings and where we're at right now, a determination of any risk of C. continensis and to identify areas of potential risk. This is where definitely a risk assessment is going to be able to be done, or at least determine any risk. But I also am very cautious of doing something like that because there's so many data gaps. None of the actual outbreaks or the infections, illnesses that have been reported have been associated with water, and so there is a definitely, I'm really cautious of pursuing that venue. I know in many ways if we don't do it, someone else will, but yet I think it's a decision that we're going to need to make if we want to go down that route or not, because I think it's when you're doing a risk assessment, especially when there's a lot of data gaps, you're drawing from other areas to try to fill those input areas, and it could be concerning. An assessment of incidence of C. continensis infection among these communities, that is definitely something that we can based on our numbers, right? We can look into further research should be conducted to determine similarity. Again, dealing with the genotyping between those. Hopefully we'll be able to do that. Work with industry to raise awareness and precautionary measures to reduce the potential for cross-contamination of fresh produce by infected persons. That is definitely, I think, an area that needs to be addressed. Because we know it's in the community, we know the community employees are working in those industries, and we know it's a human pathogen, it's an intestinal tract infection, so that's how it's going to occur. And then finally, this was an interesting one. One of the 3 wastewater treatment plants released their water into a watershed. That watershed, the water goes to Mexico. So yeah, and that's pretty close proximity once you look on the other side of that border there. There's a lot of produce that's occurring on that side.
So just, I'm sorry, going back to the issue of those illnesses. The people who got sick didn't travel, but we don't know necessarily whether or not the food was imported and consumed in the United States. Just
Correct.
Imported. Okay.
Yep. Correct. And many thanks to our collaborators, our team, everyone from Walter, Dr. Gerba, Cali, Paula Rivadeneira, up in one of the regions, Masaki Kitajima, that really facilitated and helped us with our TACMAN probe assay development. And then I can't say enough of Daniela, really doing the grunt work and making sure that all this work is put out. We recently brought on Monique, and she's been really helpful, did a lot of our stats for us. And then our undergraduates, everyone from Caitlin and Gloria, actually Gloria's in the room, she just graduated. Physiology, got accepted to public health masters. And then we have Saida and Caitlin. And Saida got accepted to an accelerated master's program through MCD. So, and then many thanks to CPS for the funding that they provided. They were really forethought in terms of funding something like this, 2 years previously, before it actually we had that really large, outbreak last year. And then the Western Alliance to Expand Student Opportunities. It's an NSF grant that really allows then our students to receive a stipend while they're getting exposed to doing some of this research. So thank you guys.
Any questions for
Any
Dr.
additional?
Lopez?
Just want to make one quick comment because we work in the same realm, except different things. Your real challenge, the way I see it, is distinguishing DNA present in effluent versus like if I'm a grower, I'll ask this question: you detected the PCR positive. I mean, how do I know? Is it a spore or it's the DNA that came into the effluent, actually? So until you have a model, or whether it's doesn't matter what, some model, something to show the infectivity. So you can take lettuce or something and feed it to some animals and reproduce the disease. That's a clear indication that you are dealing with infectious entity.
But you have to have a host. That's the
That's the challenge.
That's the challenge.
So,
Really
so
good
do
point.
you have any extension or other brochures that are given to the industry just as an awareness type of thing?
Yeah, that's a really good point. Then we've been working with a group because of the outbreaks that went ahead and occurred. So there was a group that was formed. There's subcommittees. We were leading the testing validation group, and from that, then we're developing a fact sheet. Okay. And so we're going to go ahead and again, but it's a partnership with CDC, FDA, industry. We're all working together to go ahead and develop that. It's in through the approval process right now, and so hopefully we'll get that done and get it out there, so that way they can at least. And then the 19B ban method is a really good foundation to sort of start moving forward, and as the mitochondrial DNA, or I know the FDA right now is also validating a water study. So that's going to be interesting to, for it to come out and, work with that. So. Anything else?
Any other questions? Not, thank
All
you,
right.
Jerry, for
Thank
such an
you.
informative and such an
Sure.
interactive presentation. We learned a lot about
Thank you.
Cyclospora today.
Good,
Thank
good.
you all for being here today. So since, June and July are summer months, we probably will not have a food safety consortium meeting. We'll reconvene again in August and Debbie will get back to you. Thank you so much.
Thank you all. Thank you. Appreciate it.
Thank you, Walter, for coming. Hopefully we can, get back on more segments.
Yeah, hopefully I'll do it more often.
And I hope you can come.
Thank you, that was great. I'm sorry, good stuff.
Oh, I think Debbie already sent out the date, October 17th. Yes, is the right. So give them on the calendar.
My day job is, I work for
Yeah.
Stop the recording, right? To do this.